So I want to teach you about vesicular transport and ultrafiltration. David7sensitheatre@gmail.com. You can contact us and consult. Okay, so that is it. David7sensitheatre, the good YouTube channel. You can have all science about human biology and more are coming. Yeah, more about science. Okay, so don't forget to subscribe to our channel. Yeah, click the bell for more notifications. Every day we upload videos. You like, comment, and share. Okay. So vesicular transport. Uh, there are mechanisms which are involved in the transport of macromolecules such as large protein molecules. Okay. This can either pass through the membrane by diffusion or by active transport mechanisms. Therefore, the vesicular transport mechanisms will include endocytosis, exocytosis, and transcytosis. So endocytosis, exocytosis, and transcytosis are the three important mechanisms of vesicular transport. We're going to talk about each and every one here, but the major point is that these molecules which are being transported are large molecules, and they can either pass through the membrane by diffusion or by active transport mechanisms. Okay, so in endocytosis, as you can see, this is a macromolecule. This one, it comes here. It is being engulfed. Now the phospholipid bilayer breaks off to form a vesicle. This is a vesicle which transports endocytosed macromolecule. Okay, so this is endocytosis. There's fusion of non-cytoplasmic sites. Okay, yeah, so let's continue. And if you want to talk about endocytosis, it means this is a process in which the substance is transported into the cell by infolding of the cell membrane around the substance, and then the substance is internalized. So this endocytosis is further categorized into three types. It can be pinocytosis, when the substance is in liquid form; phagocytosis, if the substance is a little bit of solid; and receptor-mediated endocytosis for large molecules where the endocytosing requires a receptor. Okay, so pinocytosis. In pinocytosis, there is engulfing of liquid substances by enfolding of the membrane of the cell. But we can call it cell drinking, okay? Because liquid substances. For example, in the renal tubular epithelial cells, there is reabsorption of liquid substances by pinocytosis, okay? Phagocytosis, this you can call it cell eating, and it is a process of engulfing of solid particles such as bacteria, dead tissue, and foreign particles by the cells. Okay, so the process of phagocytosis involves three steps. We have attachment, engulfment, and degradation stage. So these three stages occur during phagocytosis. Attachment of the bacteria or solid substance on the cell membrane. The substance will be engulfed by enfolding of the membrane, and the degradation by lysosomes. Okay, so let's continue. Let's talk about receptor-mediated endocytosis. In this process, the substance to be transported will bind with a special receptor protein on the surface of the cell membrane, okay, on the cell surface. And the receptor protein substance complex will be engulfed by the cell membrane by a process of endocytosis. Okay, and here transport of iron and cholesterol into the cells occurs by this mechanism. So this is a good example you can give, that iron and cholesterol, they are endocytosed by binding on receptors on the cell surface. Okay, that's how it happens. So now we tune to exocytosis. In exocytosis, substances are being expelled from the cell without passing through the cell membrane. This is different. We are talking about vesicular transport, okay? So they don't pass through the cell membrane, but they are just vesicles which are fusing and unfusing, okay? So in this process, the substances which are to be excluded are collected in the form of granules or vesicles which move towards the cell membrane. So they first collect this material to be excluded, and it will be collected in the vesicles. The vesicles get closer to the cell membrane, and their membrane then fuses with the cell membrane, and the area of fusion will break down to release the contents, the exterior, and leave the cell membrane intact. For example, if you look at this, collecting substances within the vesicle gets closer to the cell membrane. This is intracellular fluid or cytosol and the like, and this is extracellular fluid. So there is fusion of the cell membrane with the vesicle because all of them are surrounded by a phospholipid bilayer, and this vesicle will cause exocytosis when it fuses. Okay, the cell membrane remains intact. Okay, so here we have the cytoplasmic sides of the two membranes fusing. Okay, yeah, I'm sorry, excuse me. So in exocytosis, I can give an example for like releasing of hormones and enzymes by the secretory cells of the body. Okay, that one of the differences you can note between exocytosis and endocytosis is that exocytosis will require calcium ions and energy along with docking proteins. Calcium will be needed, which this enters from extracellular fluid and it enters into the cell to cause fusion of the vesicles with the cell membrane. Okay, so calcium will be required, unlike endocytosis. So now we switch to transcytosis. When there is vesicular transport which is occurring just within the cell, we call it cytopempsis or transcytosis. So cytopempsis is the same as transcytosis, and it is quite similar to exocytosis and endocytosis. And the basic key steps involved in transcytosis is that the vesicle will have to be formed, have to be transported within the cell, and there should be docking within the cell. Okay, so other transport processes. All along we've been considering transport across the cell membrane, i.e. movement of substances between ICF and the extracellular fluid through the cell membrane. In addition to this, there are many situations in the body where transport of substances occurs through the epithelia and capillary endothelial cell membrane. Okay, so this is between layer of cells, okay, from one layer to another layer. Okay, so this we have ultrafiltration, and we have transport across epithelia. Okay, so let's talk about these forms and continue. So the transport across epithelia, there's movement of substances from one side of the epithelium to the other. The transepithelial transport occurs in body cavities lined by a continuous sheet of cells, such as gastrointestinal tract, the renal tubules, pulmonary airways, and other structures. So, for transepithelial transport to occur, the cells need to be bound by tight junctions and have different ion channels and transport protein in different parts of their membrane. So you get this point. Tight junctions must be there. And different ion channels and transport proteins are present in different parts of their membrane. So that's when we have transepithelial transport. And this is common in body cavities lined by a continuous sheet of cells, such as the GIT, renal tubules, pulmonary airways, and other structures. Let's talk about ultrafiltration. Ultrafiltration refers to occurrence of dialysis under hydrostatic pressure. Okay, so ultrafiltration will occur at the capillary level within the body. Blood capillaries have filtration under pressure. Okay, and the capillary blood is under hydrostatic pressure, that we know. And the pressure is around 35 millimeters of mercury near the arteriolar end and gradually declines to around 12 millimeters of mercury towards the venous end of the capillary. Okay, so through the capillaries there occurs ultrafiltration of all the constituents of plasma except the proteins into interstitial spaces. Therefore, ultrafiltration will play an important role in formation of the body fluids. Okay, so it plays an important role in formation of body fluids because the formation of interstitial fluid, the fluid which exists between cells, will require filtration of blood to give out plasma without proteins. Okay, so that is it, that the fluid within blood will be filtered into the interstitium and the proteins remain in blood. Okay, so these pressure changes between the arteriolar end and the venous end are responsible for ultrafiltration.